Good morning students, welcome to LEELA'S tutorial. So in last class we have discussed about composition and components of the animal tissue culture media. So today we will see how you are going to prepare this media. So first here when you are preparing the media, based on the type of cell you want to culture, you are supposed to select the media. So here we have different types of media, that is EMEM media, that is Eagle's minimum essential medium, or DMEM medium, which is Dalbecos modified Eagle medium, and the other medium you have is RPMI 1640 medium, that is Roosevelt Park Memorial Institute 1640 medium. You have many other media also which you can select based on the cell you want to culture, right? So first when you select some media, that media, the components, everything must be first weighed accurately, right? All the components, yesterday we have seen the basic or the minimal media consists of antibiotic, vitamins, glucose, amino acids, serum, buffers and inorganic salts. So all the components must be weighed accurately and then they must be dissolved and made up to 1 liter in double distilled water. The water must be autoclaved prior, right? So here after making up the complete solution to 1 liter, now you are going to use the laminar airflow chamber. So this is the working place where you are going to inoculate your cells and do all the further work, right? So first the laminar airflow chamber must be surface sterilized. So surface sterilization using liquids, it is nothing but your 70% ethanol. So 70% ethanol you are going to take it in cotton and wipe complete laminar airflow. Then you are going to radiate it. That is sterilization by radiation. That is you are going to switch on the UV, right? After 15 minutes you are going to switch it off. Then on the laminar airflow, or you can on the flow of air through HEPA filters, right? So once this laminar airflow is ready, now you are going to use, or you're going to sterilize the media. See, now the media consists of all the heat labile compounds. Heat labile means the substances or the compounds which are sensitive to the heat, right? So if the substance like amino acids, proteins, serum, everything, these are heat labile. That means when you subject them to the high temperatures, the proteins or the substances will get denatured, right? So in order to avoid the denaturation, here we are not going to use any heat sterilization process. Instead we are going to make filter sterilization apparatus, right? So here in the filter sterilization, here you can see the filter sterilization apparatus. So in this apparatus you can see first you have the funnel which is fitted to the flask, right? Again to the flask you have a suction pump. So this is your filter sterilization apparatus, and main thing is use of your nitrocellulose membrane, right? So here in the funnel you are going to pour the media which you need to sterilize. Then you are going to on the suction pump. So due to this suction, here with the pressure, the media which is present in the upper funnel will be pulled down, right? So now you can see drop by drop all the media is getting sterilized through nitrocellulose membrane. What is the pore size of nitrocellulose membrane? It is 0.2 micrometers, right? So 0.2 micrometers, why we are using the same pore size? Because it is the size of very smallest bacteria. So the smallest bacteria size is 0.2. So here when you pass the media through this nitrocellulose membrane, you can filter out all the bacteria, fungi or any other contaminants, right? So now you get the sterilized media. So once you get the sterilized media, right? So everything is done in laminar airflow. So that sterilized media has to be poured in animal tissue culture flask, right? So these are the flat bottles in which you are going to culture. Why we are using the flat bottle? Because all the cells are anchorage dependent cells. So here the anchorage dependent cells means they get adhered to the substrate. So here this is the substrate. Substrate is nothing but the container to which they are going and getting adhered, right? So here you have the cells which you have inoculated, the selected cells. It can be your animal cells or any mammal cells. So here after inoculation you are going to go for incubation, right? So inoculation and incubation under favorable condition, that is where the pH is maintained at 7.4, temperature at 37 degrees centigrade, the pH, osmolarity, everything is maintained. The main property, yesterday in last class I told you that they require CO2 incubator, not the normal incubator. So in case of absence of CO2 incubator, you can make use of NaHCO3, sodium bicarbonate, right? So here when you inoculate and incubate, you can see the cells start growing as a monolayer, right? That means they occupy the complete flat surface of the container. They grow as an uniform single layer. Hence they are called as monolayer of cells. So here when you observe this flask, right, this flask consists of a cap which is having the filters, right? Again these filters are used for exchange of gases. So here the filter, though the cap is closed, the filters will help in exchange of gases. What gases? That is your CO2 and O2. The CO2 incubator, when you are incubating, the CO2 will enter inside and the excess O2 will be released outside. So like that there is balancing of your CO2 in order to maintain the pH and osmolarity of the media. So once you get the monolayer, you can't visualize this layer of cells. So what you are going to do? You are going to visualize these cells by using your inverted microscope, right? This is different from your compound microscope or the light microscope. So here the light will be focused from the top and you will be visualizing from the bottom. The lens are present at the bottom, whereas in normal compound microscope, light focus will be in the bottom and you will be visualizing from the top. The lens will be on the top. But here it is an inverted microscope where the light source is on the top and the lens for the viewing is from the bottom. So here, so that you can visualize the monolayer of cells which are adhered to the substrate. As these cells are anchorage dependent cells, they have adhered to the substrate, that is the surface of the container, right? So now these cells you can visualize by using your inverted microscope. So once you see the monolayer, what you are going to do? You are going to again go for the subculture process, right? So in order to obtain your cell lines. Okay, if you have any doubts, put it in comments, and for further videos please subscribe.